Welcome to Year 10 Biology class. So in this class, for 4 weeks, you're going to learn about the structure of the DNA. So started from what makes the us, basically. I know last week, those who did the chemistry, I told you everything us we're made with the atoms, but it's slightly above their level than atoms too. So we're going to learn about how we are structured, why it simply think that's this, why you look similar to your parents or not. Okay, if not, you have to question about why not. Why am I not look similar to my mother or father too? So you're going to learn about the genetics in here as well too. Why you're sharing the same traits as your dad or mom. Is anyone color blinded? No? Okay. Well, no, I'm not. Okay. So if you know any someone like color blinded, or have you watched the movie The Glory? Yeah. 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 It's a very important clue that there's a daughter of the bad guy. Yeah. It's kind of telling you about why the gene's so important as well too. So it could be quite interesting. At the end of the modules, you will learn about some gene technologies, they include some of the things, ethical and moral issues. So in schools, sometimes you are asked to write the reports about those ethical and moral issues. If you're not generally in the girls' schools, boys' schools, they don't just really care sometimes. They just move on to something else. I'm not too sure whether we can continue on up to the evolutions. Okay, evolution takes about the last 2 chapters, natural selection and evolution, which is very interesting topic. Okay, if you like that these genes, DNA, and evolution and natural selections, you should consider choose the biology in year 11 and 12 because you will get very similar topics in year 12 as well, but more in details. Okay, any questions before I start? Okay, well, if you're not a big fan of those, don't worry about it. Okay, this will be the last time you're facing these DNA structures in your life, probably, unless you choose science in university. I hope you don't. Okay. All right, let's talk about the DNA then. So first of all, we need to know what the DNA stands for. On then page 2, that shows the DNA structures. Now, DNA it's a shorter name of this. So when we say DNA, it's a deoxy, okay, ribonucleic acid. From here, we just take the front letters of the deoxy, nucleic, and acid, and we name that as something known as DNA. And the capital letter like here. The easiest way to pronounce this is give it some space between deoxy and ribo, okay, and ribo that and that like here. So you can pronounce it as deoxy, ribo is the right term, nucleic acid. Okay? And then fast forward, deoxyribonucleic acid, or simply called DNA. You also need to know the full name of this, just like you have an initial, but we also need to know your full name, the spelling of that too. So sometimes you get questions about, hey, name the DNA. That's easy questions then. It simply means it indicates some of the things inside to here. So deoxy means it has some kind of do with the oxygen, and nucleic means like acid too. So don't worry about the chemical terms if you're not interested in studying biology or molecular biology in the university yet. Okay, but you need to know how the DNA structures look like. This is something very important, and that's a related little bit of chemistry. Little bit of chemistry. Okay. When we describe the DNA, we also known as their DNA structure, also known as Watson's Crick model, because those 2 scientists are known to be discover the structure of the DNA first. Okay, as you can see from the figure, that's a figure 1 in here. Do you guys see that we have one strand? It's kind of twisted. This is very important. When we say the Watson's and Crick's model of the DNA, that's known as kind of 2 strands. So you can imagine like this: 2 pieces of papers. Do I have the piece of papers? No. Okay. Sorry, grab a piece of paper. Sorry. Okay. Lucy, do you mind if I pass the 2 pieces of paper from the printer? Thank you. Okay, the easiest way to show that is you can think about the two pieces of paper like here, fold it in half, another half. That's a one strand of the DNA, okay? And there's another strand over here. 1 and 2. Maybe one more. That's better. So you can assume that in the one single strand over here, there's a various information hidden here. So I'm gonna write something like A, B, C, D, and up to E, F, G, H, I, like here. On this side over here, there will be another A, B, C, D, and so on like this. Okay. So why we have then is it's kind of interesting to think about why we have this kind of structure in our DNA structures too. So strand means that they're not like in a linear like this. They're kind of twisted like this. Okay. So what we have inside this is some sort of information written in here. When this is kind of twisted, not just that, like kind of twisted like here, what you probably do is this information here is kind of protected by the backbone of this another strands over here. You see that? So if that's exposed like here, like this, okay, there's a risk of that this information can be leaked or can be broken too. So the reason why we try to have this kind of helix, this is called a double helix structure, is kind of twisted structure like that, is due to what? To protect the information inside of the strand. As you can see from that too. This is kind of giving some kind of protection. So on the second dot point over here, DNA stored in the nucleus of the cell, where it's packaged into thread-like highlight there please, thread-like structures called the chromosomes too. Kind of thread-like. I'll show you about them, what I mean by that too. All right. So when we see that nucleus structures, this on your page, what page? Page 3. These are known as the subunits of the DNA. So when we call the subunits, the smallest unit we can find the DNA of this. Okay, you can think about this over here. Okay, when I cut this up, that's a subunit. Okay, each of this will be the subunit of the DNA too. When I connect them here like this, it makes the strands of DNA. That subunit. So subunit of the DNA is called a nucleotide. So you can think of like the small pieces of the DNA here. Okay, in each of the nucleotides, we believe that the structure look like this. I'm gonna copy that structure over here. It look like this. There's all over here, the circle over there, and has a pentagon shape of things. And we have the thing, the base over here too. Now, give each of this. This on the top over here is known as the phosphate, or sugar over here. Sugar and phosphate. And that is known as the phosphate. Yes, those who remember the phosphate, PO43 minus is the phosphate bonds over here. Little bit of chemistry, but that's it. That's all you need to know. You don't even know to the valency of the phosphate here yet. This is called we call as some bases of that too. So also given on here sugar, base, and phosphate parts over here. So what do they do? These are kind of, as I said, they are subunits. It's a small piece of the DNA over here. The base is the part that this is carrying the important genetic, we call it code inside over here. And there are 4 types of bases. We have C, G, A, and T. In fact, instead of writing like here, I'm gonna write like this: C, G, and A, and T. Yes, and unfortunately, you need to memorize the name of these 4 bases. It looks complicated. So, C stands for the cytosine. A stands for the adenine. T stands for the thymine. And the G stands for the guanine. These are the words that you never ever heard in your life too. Yes, and as I said, This is the probably last year that you will hear about that if you didn't choose, you're not gonna choose the biology for year 11. Okay, till then, till you finish year 10, you need to memorize the name of this. 4 bases, and there's the rules. The C and G goes together, kind of pairing each other. A and T are pairing each other. This is called the base pairing rules. So in the symbols, we say the C and G are making a pair. A and T is making a pair as well. That's what we believe in the base pairing rules. Now, then let's redraw that into the some DNA structure. I said this is a subunit, the smallest unit we have. I show the connection of these then bases to here. Assume that we have an A over here, the base pair A, adenine, and this backbone, this sugar and phosphate will be connected to another sugar and phosphate like this here. This kind of connected here, making the connected to another sugar, okay, and then that goes on to another phosphate, like that, and it will have a base pair of that like this here. Okay, I show that again. The phosphate end kind of comes out, connected to the sugar, and connected to another phosphate there. I'm running out of space, but okay, should be there. Okay, this, and then the base pair there. Okay, I show that there's A, C, and G base pair is kind of covered here. So this, as we can see from there, do you see that the very important information, the bases, kind of protected by this side over here? So you can imagine these informations kind of protected by these backbones. So we call these sections as the backbone of the DNA. As we all have a backbone too. Okay? And what's inside over here is a base carrying some very important information about yourself. Okay? It will be it's all related to confidentiality later on. Now, on the other side of that too, we can have another base pairs too. Now, the rules that you need to remember base pairing is A goes on to T, C goes on to G. So when we have an A on this side over here, we must have T is kind of pairing to that base pair. And then on the other side of that, that's connected to another phosphate and the sugar on the top over here too. Under here, we get the C add to the G in the base pair. And this is also connected to another phosphate and sugar like this. And this is kind of connected like here. And the followed by C to G to C. Okay, and that's going to this, kind of related like this here. So do you see like there are 2 backbones are located? So as I showed you the linear structure of the 2 DNAs on the papers, one side over here. So when we have A to G, it's kind of facing up like this in this method too. Now what you have to imagine is these are the thread like this, kind of twisting each other. The backbones are twisting each other like this, kind of all. You can just twist it like this. Yep. So what they do is, again, the backbones are kind of hiding the information inside too. Okay. Good. So that the information will not be leaked. It will be protected from some external or internal factors in that case too. That is the main structure of the DNA we can draw in the simple diagram forms. Again, each of these symbol of that is known as what? The nucleotide subunit over here. And inside of the nucleotide, we have sugar, phosphate, and the base inside of that too. That's how we draw the DNA out and how we understand our DNA structures. So on your page 4 is showing the base pairing rules again too. The base G, guanine and the cytosine C always pair together, forming the C to G pair. The base pair A and T always pairing together, forming A to T pairs like that too. Yep, it's not hard to so you can just think like an A, T, C, G in that order in terms of the base pairings. It's not really hard to forget that yet. Okay, that's how they look like. Okay, sugar and phosphate. Oh, sorry guys. Okay, it's been a while. No wonder. let's switch. If you wrote it down, that was actually sugar. Yeah, and there's a phosphate. Yeah, phosphates are working as some sort of glue, and the sugar is kind of directly attached to the base. So that's what it look like. Okay, so base pairing rule again. A to the T, C to the G. But let's revise that. A, Hayden, A stands for what? A stands for? Adenine. Matthew, T stands for? Thiamine. C stands for? Jenny? Cytosine. And G stands for Damin? Guanine. Yep. And the DNA stands for? Deoxyribonucleic acid. So I'm trying to remember as deoxyribonucleic acid. Rather you say deoxyribonucleic acid. And spelling will be, I know it will be confused after today probably. Okay. But I don't blame on you even if you forget about the spelling of that too. All right, so some genetic codes on your page 8. Then why these are the important? I should only show you that 3 factors over here. So let's think about this there. Assume that in the one strand of the DNA, okay, there's a strand of DNA over here. I have a base kind of lined like this. Let's say I have a ATG, ATCG, AGC like this over here. Okay, that's basically on a strand of DNA we have over here. Okay, according to the base pairing rules, A can be only matched to the T. T can be only matched to the A. C can be only matched to the G so far too. So assume that we have a trend over here, and what we're going to look at is we're going to look at the sections over here. 3 pairs, 3 each, 3 there, and we have a C. Let's ignore the C over there. Now, by looking at the section of that DNA over here, okay, assume that's in your corresponding to your eye color. Okay, that might be corresponding to probably your height. Okay, that might be corresponding to, I don't know, maybe like your hair color too. So each of the segments of DNA will be corresponding, which will be responsible for the some parts of your bodies too. So ATG is not the real, by the way, but ATG is kind of telling you about, like, this is some information about your eye color. So if we all have the same ATG in the one strand, another strand on the top, we must have the base pairing to those. Okay, so for the A, as I said, there's a T will be pairing, T, there'll be A will be pairing, G, there will be C will be pairing like this too. And that segment over here will be corresponding to your eye color. Now, if Matthew and me have the same code of this, ATG, in our DNAs too, that means that we must have the same eye color as well too. In other cases, like you can think about your parents, generally you will have the same hair color with as your parents, okay? Whether that be brown or black. Yep, that means that you and your parents are sharing the same code, same segments of the DNA in there too. Because later on, what they do is, this is kind of related to something called a protein synthesis, making a protein of your body. Yeah, you don't need to know about the protein synthesis in year 10 yet. Okay, but the reason why these are important is this is kind of giving you about blue kind of map of your body. So if I can take you some of the sample of your DNA or any of the cells, okay, from the nucleus, I get the information ATG. I don't know who you are, damn it, okay? I haven't seen you before, but I only get the sample of this from someone else. I don't know, maybe from black market, or I collect it from your wardrobe, wherever. And I got hey, I found the ATG. That means that, as you have an ATG segment in that section, that means that you must have the brown eyes or brown hair. Does it make sense too? And this is how we use in some criminals, like, to detect it, like, whether they are Caucasian, Asians, or like, or like where they're from too, but they're related as well too. So that's known as genetic code. So on your point over here, and the second dot point, this genetic code is the same for all organisms. Therefore, DNA differentiate not only individuals of the species, but all living things of the earth. Okay, so that's, assume that this is the sum of me. Then I show you another one from, let's say, the Hayden. I have an ATG. Okay, but instead of A, I have a, like, say, T, A, C there. I still have a G, A, and G like this. That's yours, Hayden. Okay, whether or not. Okay. Now, do you see like any difference between this and that? Yeah, the high section there. What do you see? What's the difference? Yeah, I have A started from A. You're starting from T over there too. Even though we are sharing the most of the code like this here, that means that we should have the same eye color. We should have the same like a hair color too. But because we have a different TAC, or the height section over there too, we might have a chance of a different height in that case too. Okay, and that it can be kind of inherited from your parents. So generally, your height is followed by is kind of following to your genes from your parents, or it could be environmental factors in these days. Okay, so by looking at that, can we say these two people are not identical? Okay, by just looking at one change over here too. This is one way we can differentiate individuals. Okay, now I show another trend over here. So instead of we have ATG, okay, not the eye color. We may have some like AGC like here, and TAC there. Not even the TAC, like TA, and say G and A there, like this. So what you see there is, hey, you might be questioning, is that for humans' DNA? If you question that, you're right. I'm showing some example of let's say a dog or kangaroos, which is about, you heard about the monkeys and chimpanzees, about 99% like matching to humans' DNA too. Okay, but that's kind of 1%. Let's say I share the C over here. I'm missing one over here. But even that missing, the simple missing one, make a huge difference of deciding the different species. This is showing the DNA structure of maybe chimpanzees, and that's you. You and chimpanzee only have one difference, which is G over there. Okay. In fact, us human have the very set number of something we call chromosomes. It's very set number of the DNA strands. Okay. Other animals would have a very set number of that too. But because of that, we all made with the same chemicals. We all made with the same sugar, phosphate, and base pairs too. But numbers of them, or the arrangement of them, will decide whether you're human or not. It will decide whether you're male or female. It will decide your haircuts and everything. So DNA is pretty much everything about the life. Any questions about up to here? So don't worry about this kind of this base pairs over there. So there's more learned in the university later if you want to. But yeah. All right. Then let's talk about some chromosomes. On your page there, the figure 5, could you highlight that figure? Not the dot points, the figure is very important. You don't have to. You can just remember that diagram. Just remember the diagram. So, Damien, have a look at the diagram of the figure 5 over here. Comparing the size of the chromosome to DNA, yeah, which is bigger? Yes, that's all. That's one thing that you need to remember. Then what would be the relationship between, just by looking at the figure over here, what would be the relationship between DNA to the chromosome? Yeah, can we, by looking at that thread like over here, can we actually say, hey, the chromosomes chromosome is made of the DNA. Okay, we have a long DNA kind of threaded, kind of joined together. And for the help of the proteins over there too, you can imagine inside of the kind of threading, like this is kind of protein work like. They kind of thread like this. Okay, that's us wrapping the DNA on the proteins like. And we have a collection of that kind of making structure which is called the chromosomes. So one thing that you need to remember the difference between chromosome and DNA is chromosome is bigger than DNA, first of all. Or you can say chromosomes are made of the DNA and proteins. Now, what is chromosome then? Chromosome is something that we can find in the cell's nucleus. So we all learned about cells in the year 9. Eukaryotic cells, okay, some cell walls, and there's a cell nucleus there too. Why is the cell nucleus so important? Yeah. Why is the cell nucleus so important? But think about if you destroy the cell nucleus, if you have no cell nucleus in the cell, what happened?
It wouldn't be able to operate properly.
Yep, it would not functioning, first of all. Okay, so cell nucleus in the cells are working as your brain, basically. So you can imagine about you without your brain. And that means your command center is basically collapsed, so you have nothing to control. You can pass on knowledge. So nucleus, when we think of nucleus here, so that's a cell. Eukaryotic cells, that's a nucleus. Can you see that? Okay. When we see inside of the nucleus, we can see in the humans that we have 23 pairs of the chromosomes. Something look like this. Okay, so one chromosome looks like that. That's one chromosome. When they're paired, it look like the X shape. 1 and 2, like that over here. These are pair of the chromosomes. I'm just showing the copy of this over here. These chromosomes again, when we look inside what's making these chromosomes, we can see that that's kind of they look like this. A line of proteins and the line there is kind of wrapped around with the DNAs. Like this structure over here. Again, when we look inside of the DNA, what's inside the DNA again? We have sugar, phosphate, sugar, and the base pair in the DNA structures. Again, that's phosphate, sugar, and the base of that too. So what's the relationship between then chromosomes and DNA, Hayden? Okay, let's talk about the common thing, like the similarities. Why the chromosomes and DNA is so important? As we learned about something in the code. Can't function, that's one, okay? But imagine you have no DNA, so they know they need to pass it over. So all the DNA and chromosomes, they are responsible in terms of your genetic information. Some genetic information. So that something that you receive from your parents, from your mother and the father, there's something you can pass on to your children too. So if you haven't seen your picture of your great-grandfather or great-grandmother, you probably see some similarity between you and them, your ancestors too. Okay, it's all carrying the genetic information too. The structure is simply they kind of developed whether they want to protect the information because it's so important here. So chromosomes and DNA, or even the nucleus are responsible in what? Carrying your genetic information. That's the one thing. But what's the main difference between DNA to the chromosomes then?
Chromosome made out of DNA.
Yeah, chromosome made of DNA. So in terms of the DNA and the proteins too. So we cannot say, hey, the DNA is same as the chromosomes. You get what I mean in here? Okay, this is more like a collection of DNA on the chromosomes. And when we're talking about DNA, we are generally talking about DNA sections or DNA like in strands in that case. Make sense? Okay. The same as like you are Jenny. You are like in a person like Jenny. Okay. By looking at your fingertip, I cannot say that's a Jenny. Okay. By collecting all that Jenny's information, we see like a Jenny yourself. That's you as in like a chromosomes, fingertip. Okay. That is pretty much same as the DNA sections over there too. Make sense? I'm repeating this because like you actually get questions like, hey, compare, contrast the DNA and the DNA and chromosomes.
Wait, just confirming the difference between chromosome and DNA is
Yes.
Chromosome is like made out of DNA and protein.
Very good. It's actually given on your page here in the chromosome sections there. So chromosomes are carrying the cell's genetic information, second dot point. They are composed of the substance called the chromatin, which are a combination of DNA and proteins in that case. So chromatin will be some section of that too. So when we're taking some segments of that, that's a chromatin. Chromatin, some section of that. And that's also known as a nucleotide. So biology is full of the new terminologies. And to succeed in this biology, it's a matter of like how you get used to those terminology first. And then use that word again and again. And describe them too. Okay. It's been like 20 minutes. Guys, with your spoon now, okay, can you just kind of shake a bit of like the strawberry on the top too, so you can get some more. Yes, I want to get the fluids out further. Okay, so force them to extract more of the strawberry extracts. Oh, ripped. Okay, and then leave that for another, like let's say 20 minutes. You do that for a while. All right, that's on your page 9. You will see some of the microscopic picture of the human's chromosomes there. That's actually what you can picture when you're looking in the nucleus. Okay, put it into like in the petri dish, and you will see like some pairs. But do you see that? Each of these over here, kind of like a made pair, like over here. So that's actually known as chromatids. Chromatid. Chromatid. When the chromatids or chromosomes and chromatids are kind of combined like this over here, they're all making into this pair. So in humans, it's good to remember this. In humans, we have a total of the 46 chromosomes. 46 means we have, it's not the chromatid, we have 46 of them in humans. But in pairs, pairs as in like we're comparing the height of the chromosomes, we lined it up, or we're seeing that pairs of them like here. Pairs means that divided 46 by 2, we have 23 pairs in our nucleus. Each of cells means that in each of your cells, in each of cells nucleus, whether that's from your hair, from your skin, from fingernail, your saliva, you will always carry that same number of the pairs of this. Okay. Now, other example in dogs, where there are 39 chromosome insets. You don't have to remember the numbers there, but that's kind of telling you why dogs look different to you. We don't look like a dog. I mean, we still have 2 eyes, 1 nose, mouth, and the teeth, but the structure of the teeth is different. their nerve system is quite different too. In peas, there are only be 7 chromosomes in set. Pea plants. Okay, so that's kind of giving you ideas about why having a different number of the chromosomes in the nucleus can decide the different species as well too. Or having different code numbers there, kind of code structure, can also give you a different species and the individual type. Now, when we say the 46 over here, basically I'm talking about what? The total number of the chromosomes in your body for humans. And sometimes we use a 2N, where the N stands for equal to haploid. That's simply I'm talking about what? The number of the pairs. Number of the pairs. Now, so that is an N in that case here. When you have 2N, this is known as the times 2 diploid. And that's telling you about number of the individual, all the chromosomes, the whole chromosomes. So in humans, we have a haploid number of the 23, diploid number of the 46. So I'm going to rewrite over here, okay? N stands for the haploid number of the pairs. 2N is a diploid. Haploid. So you're going to commonly use that, hey, how many haploids dogs can have then? Let's have a look at the dogs. In dogs, there are 39 chromosomes in a set. So that's telling you about what? The diploid, the haploid number, number of the pairs. Okay? In other words, in total number of the chromosomes, dogs will have times 2 of that, which is 78. So that will be the dog's haploid. And this would be the dog's diploid. Same for the pea plant. Pea plant have their haploid number of the 7. That means the 7 pairs of the chromosomes. And 14 of the what? The diploid number 2. When we come back to the haploid and diploid, when we're talking about some Punnett squares, now how did you get the genetic information of your parents later on? So when we're lining up those pairs, you will get something like this. This is page 10, figure 9, and it's showing the set of the human chromosomes. And did you realize that they'll given number 1 to the tallest chromosome from here? So what they do is they kind of align from the tallest to the shortest up to 22. Okay, so up to 22 we have some kind of the chromosome that can determine your physical feature basically, or your personality. Here, the last one, you only have either XX or XY. These two are called the sex chromosomes. And this one decides whether you're a male or female. All the males will have the XYs. So it's one shorter. It's good to remember that, oh, XY, the Y chromosomes, the Y sex chromosome is actually shorter than the X chromosome. It's good to remember this. Whereas X chromosome have the same length of the chromosome type. Okay? So that's either 1 or 2. So even for those, it's kind of sensitive topic too, the transgenders. Okay, so they can physically change the structure, the feature of the from male to female, female to male, but something they cannot change is this, because they're born with that, or if they are a male to female, they're born with the XY chromosomes. Okay, so they what they do is they can administering some of the hormone traits, so it's kind of getting some more of the kind of female-like features. But it doesn't mean that they will get the change in XY to XX. This X and XY, the sex chromosome, exists your head to toe. Every cell in your body will carry this 46 and 23 pairs of the chromosome in your body. So it means that if I take the cells from your skins compared to cells from your, let's say, the hair, they will have the same number of the same exactly identical the number as well as the type and the length of them too. Make sense? Okay. So again, 22 and 46 over there too. That's something we can say. Any questions up to here so far? Number of the chromosome, the difference between DNA and chromosome. DNA stands for that base pairing rules. We are kind of running that. So I'm going ahead of your school now. In half an hour. Okay. Any questions? Okay. All right. Let's have a short break before we move on to something over here. I want to do something a little later. Yeah, basically I talked about XX and XY, and I'll leave that for here. Okay, could you shake just a bit of like the extracts, please? Yeah, it takes about like sometimes hours to extract the DNA. Yeah, okay, you're doing good. So while we then, let's think about the experiment today. This is known as the extraction of the DNA from the strawberries. So first, why do we mash? What's the purpose of mashing the strawberries?
So it's easier to bond it down.
Why?
To extract the
Yes, that's one.
To separate the seeds.
Seed doesn't really matter for that case. Okay. Now, guys, think about like mashing. So from the one small strawberry shape here, that strawberry there, when you're mashing, you'll actually what? Increase the surface area. Okay, so instead of we have a small surface area over here, when you, the same as you bite something, you're chewing something, your food. The reason why you're chewing the food is basically you want to increase the surface area of the food so that you can digest it faster, digest it better. So mashing, as you say, we're extracting something in there and out, as well as increase the surface area of that too. That's one reason, okay? Now, we put this salt, a little bit of water, and this water comes out from the juice of the strawberries, and also the detergent, or the dishwashing liquid, the liquid. So what they do is, the dishwashing liquid is kind of separating the cells from the cell. It's kind of breaking down the cell structure. So we try to get the nucleus there. Does it make sense? By extracting, I'm putting, okay, we're good? By putting some dishwashing that too, yes, that's kind of destroying that physically, as well as by adding some of the dishwashing, the liquid, it started to kind of breaking that, chemically breaking that the cell wall of the strawberries. So once you put that and give a gentle mix there, we are start to getting what? Some nucleus out from that cell wall. So your extraction you're getting down there, those are the nucleus now. Kind of nucleus of the strawberries you're having now. Okay, salt was kind of helping to dissolve better. To increase the solubility of that too. Don't worry about this, okay? You probably do that in the university lab if you do molecular biology. And I don't recommend that. It's fun, but it's a love and hate, love and death. You will either really hate it, you will love it. Okay, and then once we get it up to here, discharging that, we extract the nucleus. And then what I'm going to do next is I'm gonna put some really strong alcohol. So once I collect all that extractions, yeah, and you will see something floating on the top. That's the kind of chromosomes of DNA. Okay, we'll see. Let's wait. Okay, suppose 3 more minutes break, and then we're gonna move on to genes. Anyone want to go to the bathroom? Lollies? Oh, where's my Skittles? Could you please open the page 13? So we're going to talk about the section, something called genes here. So for the biology, whether it's biochemistry or physics, what I recommend to understand the content of biology is use figures or diagrams first. Because yes, you can memorize some textbook definitions from your various textbooks too, but sometimes they have different definitions and it will confuse you a lot. But they will share the same diagrams all the times. So when we're talking about the difference between what the chromosomes and DNA over here, okay, we believe that DNA is what is the chromosomes are made out of all the DNAs with the proteins there. Okay, same in here. When we show the difference between something called a gene, so you've probably heard of genes, yeah, or genetic or genes. It's actually talking about the sections of the DNAs. Sections of DNA. So how do we show them visually? This is how we show that visual section. So as we can see the DNA strands over there, each of this is a nucleotide and making the DNA strands over there too. When we're taking a sections by sections and by sections, so the sections there, these are called the genes. Okay, so genes are basically what? The collection of the DNA or the sections of DNA, that's it. And it will carry, it depends on the gene section over there too, as I showed you before. Some of the genes, some of the section of DNA will carry your information about your hair color. Some of the genes will carry the information about your height. Some of the genes will carry about what? Some features of your, say like your skin color, or different features too. So yes, we are all made of this type of nucleotide, same chemical structures, but depends on the how structure and the genes are structured, how the DNA align too. Okay, or as I showed, we can have an ATG to the CTG, something like this too. So difference between the one gene to the next is the order of the base along the DNA strand. This is how we can differentiate the genetic or genes. And the number of the bases in the section of the DNA. As I showed you between from the previous example, we have like 9 DNAs and had 8 before. Does it make sense here? So you need to clearly know the difference between DNA to chromosome 2, lastly the gene. This is one question that's very common in school exams. You get a question like, and this is something that you can simply visualize it. Yes, okay. Sour. Yes, you can memorize the textbook definitions. Yes, I would like to see that in your answer in the exam preparation. But I want you to imagine about this first. This comes out first, and then you can write it about in the words. Does it make sense? Okay. So let's try that. Some of the questions we have on the page 14, 15. First here, could you guys define that? So let's do the 14. Could you guys define the DNA, nucleotide, the chromosome, and genes separately now, please?
Do I answer in the book?
Yes, answer in the book. That's yours. I get you the booklet next time because I was not able to prepare the one before, but Okay, so, Damien, how would you define the DNA?
Deoxyribonucleic acid.
Yep, it's known as deoxyribonucleic acid. This is the full name of the DNA, yeah, and
Carries genetic information.
it carries the carries the genetic information, yeah. Anything else?
I haven't read it.
Okay, does anyone want to add anything on the DNA section? Matthew, nothing?
Not really.
And molecules? That's what I read.
Molecules? What do you mean by the molecules?
And
Molecules.
molecules. Yeah.
More molecules, yeah. What about molecules? Molecule means that two atoms are joined by the chemical bondings. So anything from you, Hayden? About DNA? Yeah, it carries genetic information. So what makes the DNA? What's inside the DNA? We will have the, it's made out of something we call a nucleotide. So that's given the function of the DNA, what is the structure of the DNA then. Then let's talk about nucleotides. Matthew, can you tell me about the nucleotide then?
It's separated. It's like a DNA that was cut off.
Separated, okay, separated. Or shall we use something called this subunit of the DNA? I like the separated though, okay? And?
Alright, how it consists of the sugar,
Yep,
phosphate, and
Sugar.
base.
and the phosphate. So let's write it like here. Sugar and phosphate is something we call the backbone again. So backbone giving the backbone structure, and the base pairs. And tell me about the base pairs again.
It's AT
Uh-huh.
and
We have a 4A adenine, T
diamine,
thymine, C
uh,
cytosine, and G
guanine.
guanine. Yeah, I'll make you to expose to those again and again and again and again. This is like, imagine that you're facing that in the school exam, like, oh, name those ATGs. You'll be like, oh, oh. So just make them yours. Try to practice it again and again. I know it's hard. I know you don't like it. You don't like them. Neither. I don't like to see them though. But things happen. I hope you get 100%. Ask your sister what she got for your biology last time. Oh, she did pretty good. Yeah. Oh, she kind of aced it at the time. She would say, I don't remember though. Okay. Ask your sister too. She did pretty okay. Oh, she did actually pretty good last year. Yeah. I hope you guys get the same test paper, by the way. All right, next one. Chromosome.
Chromosome is made of DNA and protein.
Yep. So it's made of the DNA plus the protein. So can we visualize it again too? It's a bunch of proteins there. It's kind of wrapped around with the DNAs like this too. This is something that you can use. You can use to imagine about chromosomes, but also the shape of this. And what else? Small function of the chromosomes.
Carries genetics.
Yeah, it carries the genetic information about yourself or about ourselves. Anything else? Jenny? Chromosomes?
It's located in the nucleus.
Yes, located in the nucleus. Yes, and then let's go for the humans. In humans, how many do we have?
Right. There's like 76.
Something 6, yeah? Yeah? 106? 96?
No, you said no. Sorry, 40. No.
You're talking about different species, are you? 46 chromosomes,
And
okay?
23 pairs.
Okay, chromosomes and 23 pairs. Shall we use the fancy word there? 46 diploids and 26 haploids. Keep using that terminology again and again. Biology is all about the terminology. I know this is why some people hate it. I didn't like it until I finished uni. Yeah, well, it is what it is. Suck it up, okay? You will get those questions. What can we do? All right, the last one, gene. Damien, tell me about the gene.
The section of DNA
Yeah.
arranged along the chromosomes.
Yeah, section of DNA along, oh, I like that. Along with, sorry, say it again. Along with?
Along the chromosomes.
Along the chromosomes, okay. What's the function of the gene?
It carries like your, I think it carries your genetics from your parents.
Yeah, it carries the genetic information. Again. So do you see that? What are we talking about? What genes, chromosome, or even the DNA? They share this. Yeah, they'll have only one function. They carry the genetic info about yourself. So genes more about chromosomes more about collection of those genetic information, except strands of them to many strands. Genes are more specific about what information you have. So it's more specific about your hair color, more specific about your height and skin colors. By looking at the chromosomes, it's hard to determine like your height, your skin color from there. But by looking at the gene, having the different base pair, the base pairing, yes, it can tell. DNA is simply a subunit of the genes too. So it's made of the nucleotide like here. Make sense? Okay? So you will get the questions like this. So it's on your page. Let's skip some of the pages there. Page 18. That's the kind of questions like I got it from one of the school exam questions before. Compare a gene and chromosome. 6 marks. That's all the gene and chromosomes there. So this, no, I know you will easily get panicked by looking at, oh, aren't they the same? Aren't they like, what's the difference? Started from the visualization, started from the diagram like here. Much better to use that diagram. You don't have to draw it in your exam, okay? In your in your answer sheet. Okay, but if that helps you to answer the question, draw it, even though it's not marked. The markers we're looking up your working out too, so we can determine whether you understood the contents or not. Don't just start to jump in without understanding, without visualizing. This is very important in studying biology. That's the chromosomes, and this is a gene. Okay, if we can start from, hey, the chromosomes are actually what? The collections of the DNA and the proteins too. And the genes are only talking about sections of DNA along with the chromosomes. Yep. So let's compare means that you need to talk about the differences between the gene and chromosomes. So directly comparing both, one is about like collection of the DNA and plus the proteins in there, which makes them what? It also carries the genetic information. We know that too. Located in the nucleus, and it has 46 in the humans and 23 pairs in the haploid and diploids. Whereas genes are more specifically talking about the sections of DNA. It's kind of telling you more about you. Chromosome itself is probably telling you about your species, having the same number of the 46 chromosomes in your nucleus. Okay, but it doesn't tell you, by just looking at the one chromosome over here, it doesn't tell you what you are. It doesn't tell you your features. Does it make sense? Okay, so by just having a one chromosome and looking at one chromosome really doesn't really tell you individual traits of you or individual features. Genes, by looking at the genetic codes or genes, sections of DNA, we are able to get what? Your individual traits and the features. Does it make sense? This is how we can compare the genes and the chromosomes. Do you want me to repeat that again? Can you answer that then?
Oh, yeah.
Yep. You never know. You're gonna have the topic test in 2 weeks. Huh? What? Yeah,
2 weeks?
2 weeks. Yeah.
Yeah?
Yeah.
Really?
Yeah. Then what? For chemistry? Oh yeah. Well, I
No,
forgot.
no, 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 no.
I have to give you the questions about chemistry too. Guys, I can make up the question. I can put it up in your topic test whenever I can. Okay. Don't worry about the chemistry for now. Okay. We could probably coming back to chemistry in term 2, in the middle of term 2. I know. But you know, you guys could start with the biology, so we have to start with that. Okay. Physics. I talked to my Saturday class and the year 10 class. I told them that you will either, once we started physics, because they're starting physics in 2 weeks, they're like, you will either hate it, or like you'll be like, don't like it.
Wait, there's no like
Physics.
I like it option?
I never seen someone like, oh, I like physics. I never ever seen. There are some people saying, oh, that's interesting.
Oh.
That's different to I like it, you know. It's either like really hate it, really hate it, so you don't want to see that again, okay, or like you don't mind it. But some people are like get fascinated about like how those vectors. So if you can get up to that stage, I strongly recommend you to do physics in your year 11, because contents you're learning now, by the way, for year 10 will be very similar in year 11 too. So this is a good test to which subject you need to choose. You need to fill it up your subject selection in term 2, by the way. Started from term 2, finalizing by end of term 3.
For the question, if it's worth 6 marks, do we have to do like 6 lines worth?
No, it should be like 6 different lines. It'll be like the difference between the, first of all, in terms of structures, that's 2 marks. Difference between like a structure difference between chromosome and gene, that's 2. Okay, there are functional differences. Okay, and let's go up to 3 to 3 in that case, because you need to define what the chromosome is. You need to define what the gene is correctly.
So do we just talk about similarities and differences?
Yeah. So to get the 6 marks for that, okay, I'll give you that the guideline then. Define the genes and chromosomes separately first, okay, and then compare their structure. In terms of what chromosome made of the DNA and protein, genes are sections of DNA along. So there you have to compare them so that you can say what chromosome is more collection, the more bigger term. Then there's sections of the DNA, which could be like it's not a subunit, which is just telling you about sections. And then for the next 2, you have to talk about what there are functional differences too. The functionally chromosomes will kind of tell you about what kind of species you are by looking at the number of the chromosomes inside the nucleus. Okay, but it doesn't really tell you about what your very unique, your unique individual feature. That's what we can determine from the gene. Okay, so if you're watching like dramas like the CSI, they're saying like they're taking the genes out, not for the chromosomes. Okay, so in class exercise 1.2 on the page 15 will help you describe the relationship between DNA, chromosome, and genes and words in the pictures too. Okay, I leave that up to you for your homework for next week. Explain your words means by the complementary base pairing, ATGC. Okay, once you finish that, could you try to do the 1.3 on your page 16? Both all of the A, B, C, D, A, B, C there, please. Page 16 and 17. Okay, while you're doing that, we'll collect that, samples.
So do this part too.
Yes, please. 1.3. You want to take a lot. Finished? No? Okay. Give me 1 more minute. Yes. You're not too sure. Okay, I'll show you that. 1.3. Construct a diagram of the complementary DNA strand for these two examples. So what you see there is you see some strand of DNA. So you can just think that as kind of things like this. A strand over here. We only have one strand. So we're missing another strand bottom there. Simply remember the rule, the base pairing rule. A adenine goes to thiamine, and cytosine goes to guanine here. So A to T, C to G. So what we have from here, A must have the T facing there. T would be A, and C, G, A, T, C, C, A, and T. And just need to connect them into a nucleotide and the DNA structures like this. Easy. Okay, same for this one. T we have A, G, C, T, G, G, C, A, C, C, G. So go like that and draw the structure of the DNA beautifully. Okay. Next one, question A again. Identify the mistakes the following section of DNA. Any mistakes? Matthew, any mistakes in here?
The G must be a pair with C.
Yep, so that section over here, that is either this changed to C or that to C. It must go to C on the, one of the side there too. Anything else?
No.
Good. Next one, B. Jenny, any mistakes in here?
The T and the C.
T and a C?
It's supposed to be G.
Yep, so we have a T and a C over here. That's supposed to be, it's either C to G, or the T to A. It's either that. Good. Damien, last one.
The 2 A's.
2 A's in the
Left
in the left hand side there too. So it should be A
T.
to T or T to A to T. Doesn't really matter.
There's one more.
There's one more.
There's one more. Good.
A
A and C, good. So instead of the A, we should be G or C or T. Good. That's the last one too. Easy. About the base pairing rules too. The hardest part is probably the naming them. I don't know. Yeah. So what you have to do is keep pronouncing them. Keep pronouncing adenine, guanine. I know that sounds stupid. It sounds like something different. But if you pronounce it better, you can write it in your exam time as well too. Make sense? Okay. Now. This is kind of, we're still collecting some, but I kind of stopped it. So, and in about 1.5 hours, we collected this much of the DNA extract. Again, for the DNA extraction experiment today, you need to know why we're using such a chemicals, for what reason. So it's a given on your page, the new page I give. It's actually in the last page in your Matthew. I kind of answered that question at the back over here. So why do we use the dishwashing liquid again too? We want to open up the cells, the strawberries, release the DNA. That's one. B, why do we use the salt? Then we put the table about like a spoon of table salt. Ensure that proteins in the cells are kept separated from the DNA. Do you remember that chromosomes are made of the what? The DNA is wrapping around the proteins. We want to get these DNA out of the proteins wrapping. What does alcohol do? This is something that we're going to do now. Now I have some very strong alcohol. This is not strong. This is isopropyl, which is something used for your hand sanitizer. So you can never smell that, by the way. Okay, pour that over here, and you'll start to see that. Do you see layering on the top? I put a little more of the alcohol on the top here. So you see some of these get some strawberry extract getting some precipitated down in here. Some things I start to clean up. So I give it a chance to shake a bit, and it looks like it. I have a bit layer of the alcohol and that over here too. Okay, I'll leave that for a sec, and I'm gonna filter this out. And what you probably see by doing that here is, looks a bit disgusting, but you started seeing that something is climbing up on the top. Climbing up on the top here. Okay, I show you that to you. There we go. There's something clumsy on the top there. Do you see that? Okay. That. Take my spoon. That is the strawberry DNA. Yeah, I know it sounds like nothing, but it's actually this kind of sluggy. Yeah, that is a DNA from, it's a punnet of the strawberry you use. We only got this much. It's still extracting some DNA there. You're seeing that comes out from like this too. So theoretically, if you have a good magnifying microscope, electron microscope, we should be able to see some structural DNA from there too. Why do we use an alcohol? So alcohol is, when you're mixing the alcohol with the water, sometimes they're not really mixed together. So alcohol is known to be insoluble. It's not really dissolving the water. So that clumps together and becomes a visible. DNA is not soluble in the alcohol. This is very important. All the DNA actually is kind of quite soluble in the water, as we are made of the 70% of the water too. Okay, but by putting the alcohol over here, you're seeing that as they're not really dissolving the alcohol, you see the clumsy, this kind of DNA structures form too. Okay. Yep, that's what you get, the DNA structure. So what we can do, we're gonna filter this out, weigh that, okay, and compare that to the weight of the strawberry, so we can determine how much of the DNA we can get out of that too. It won't be accurate experiment because we haven't collected all the extracts yet, and it'll be varied. It actually takes about a few hours to do so, but we don't have that much time. So I probably leave that, and I leave that in the filter paper so you guys can see that what happens to the DNA next time. Another thing is, someone actually asked me the questions in last year. Someone asked me can we eat that? Yeah, like I don't know why. There's some people are always thinking about, oh, can we have that? Can we eat that? Can we eat that? When I say acid and base, can we eat that? You never take to your mouth something in the lab. Never, ever. Theoretically, yes, but it would taste awful. If you want to show me that, I can share a video. Someone actually tried that DNA extract from the same experiment. He kind of vomited it out. Okay, and this had a full of the dishwashing on the liquids too, so you would not ever want to taste that. I know the colors looks tasty, kind of jelly kind of taste. Sorry, jelly kind of color, but it tastes not like a jelly. I promise that. Okay, all right. Then let's see the last section over here. Page 20 is about DNA replication. Something that we're going to do next week for the chapter 2, for when you're talking about the cell divisions too. But DNA replication, okay. Let's just think about here. Why do you think, first of all, the reason why we have a double helix kind of twisted structure of DNA is, I told you that the backbones of DNA, this backbone is kind of protecting the information inside in here. If there's something wrong, we're going to talk about it in a second. What if it's something wrong with your information here? Massive things can be happening in your body. Even one base pair, yes. Now, then why do we have 2 strands? Not only one. We never thought it that way, yeah? Why do we have 2 strands, not only one? Let's assume that, I'll show you over here. It's better use the diagrams given in here. In step 1, assume that we have a 2 strands. There's a strand 1 and strand 2 over here. Okay, when the cells are dividing, this is something that we're going to learn next week too. I'll give you some spoiler. When cells are dividing, we have a cell process called a mitosis. Have you guys heard about mitosis in year 9? Okay, means a cell division. This is good in the purpose of the growth and repair. We want to have the copy and paste, copy and paste. So I want you to think about the copy and paste your, like say, your essay probably. You're copying a paragraph, paste that basically. Similar things happen in your cell structures, your DNA structures over here. Now, in the replication, all the DNA copy, the straight one here, which is this one, is kind of separated. That's distributed as strand 1. Okay. Now, what you see there is there is something called like DNA polymerase, or there's some kind of helper or DNA forming one. So you can think that there's DNA builder comes along, and that's try to build that on this side of the strand 1, as well as strand 2 over here. So what that means is, in the chromosomes in the human body, we have a 46 chromosomes. Yeah, okay. We can copy and paste that 46 chromosomes in exact identical. There will be 92 chromosomes at one time. Why? The benefit of having a double strand is, one will leave the copy. This is a copy. Okay? 2 will be the original on that side over here. So at the end of the, we call that replication, repeat, or the copying and paste, we get the 2 identical strands of the DNA like here. Why do we important? So you know the mitosis are from the 1 cell, 2, dividing into 2. Okay, from for 46 over here, to get the identical cells for the daughter cells, they must have the 46 and 46 here as well too. We cannot divide the 23 and 23 for the mitosis. Each of the daughter cells will have 46 and 46, and they must be identical. This is a very important term, guys. They must be identical. You cannot have someone else's cell. You cannot have 45 instead of 46. They must be the 46 there, and with the exact same genetic code you're carrying right now too. Otherwise, assume that you are changing your genes every single time you get dividing your cells. At a certain stage, this could be taking a year or a month, you will no longer be yourself now. But we never thought that in that way, yeah? We always think like, 10 years ago, Damien is still Damien. I know you're grown up, okay? But you'll still be a Damien, okay? Same for me, like 10 years, 20 years ago, 20 years later, okay? I will still be me. Because I will be still carrying same information as I did, as I was born with. Cell replication, the replication is, therefore, it's important in that sense. Again, double strands has the benefit of what? Creating two daughter cells, or two copies of the one, from one. Otherwise, if we only have a one strand, we cannot make the two copies. Because if we have a two double strands kind of wrapped, and then the double helix forms, they should be able to get the two identical cells like this. So what are the steps for that too? Yes, it's important to know about the steps, but I'll probably could talk more about this next week in the chapter 2. Let's go through it quickly. Step number 1, in the first step of DNA replication, enzymes unzip the paired strands of DNA somewhere along the DNA molecules. Each unzipped section of DNA serves as template to start the building a complementary strand of DNA. So, start from unzip, from here, unzipping. We get the two strands ready to get copied, okay? And then step 2, enzyme called, yes, you can highlight that, please. You can enzyme called DNA polymerase, aka DNA builder. DNA builder comes along, DNA polymerase comes, adds a matching nucleotide to the exposed. So what they do, when the DNA polymerase comes along to it, it's kind of building that according to the base pair over here, going like this. The same as the kind of zipping your zipper. Okay, does it make sense? From the one strand, having a double zippers like this. So DNA polymerase just like working as a zipper. You're like zipping up. Does it make sense? Okay. And the step 3, two identical DNA molecules, each one of the double helix, they are also identical to original parent plane. So the key word for that is identical. They must be the identical because you are exactly copying and pasting. I mean, thinking about copying your paragraph from your essay, you're expecting them to be exactly the same or identical. Otherwise, what's the point of the copy and paste? Yep. So same as that. So they, after the replication, replicate means we get copying and paste basically. After replication, what we get the same two, from 1 to 2 identical DNA strands ready, double helix ready. So they are ready to go for what? Some cell division from there too. Make sense? Okay. And this is the reason why the DNA, we believe that, we kind of studied that the DNA kind of wrapped it, kind of making double helix structures like this, and why does it need to be doubled as well too. This is what we have been studying so far in current science. Okay, now let's talk about then what goes wrong with your body if you have something wrong with your base pair. So, assume that you have a base pairing like this. Let's say ATGCATCAG, like this here. Okay, so you have the base pairing of that side over here. Now, have you guys heard about the skin cancer? You all know about the skin cancer, yeah? Okay. Now, what causes skin cancer?
Too much exposure to sun.
Sun, specifically?
The ultraviolet.
Yeah, UV light. Okay. If you're studying physics in year 11 and 12, you probably learn more about details about UV light too. So you can just think about, we use UV light to sterilize your cups or some utensils sometimes in hospitals or some in the house. Okay. If you haven't seen like sterilizing with using UV light, so which means that UV light is so powerful, it can kill the bacteria. Yeah. UV light is so powerful. So I showed the UV light. It can penetrate through to your cells, to your nucleus. It can damage one of your base pair here. Now, let's see what happened then after this C get damaged. So we don't have C anymore because it get damaged. But this cell, this strand will still be what? going on the replication. So T, C, and we used to have like a T over here, so it's C to G over here. Now we're missing the C because of the UV light. Okay, and then the other things we can have it like follow T, A, G, T, C like this here. Now from the double helix structure here, remember the replication has the purpose of producing two identical strands. But once this one's get damaged, what happened? You will no longer get the C here. This side, yes, you will still get the normal replication, same identical shape again here. Then this cell, when this kind of DNA trend or genes, genetic codes are kept replicating, you can develop something called a malfunctioning skin cell. So it's exists as a cell, but it's not working as a cell because it's missing G over here, missing C over here. That's known as a cancer. So any form of cancer cells, that's due to many cases that due to that damaged DNA, damaged gene, damaged chromosomes too. And there are many the various factors you can have a different altered the DNA too. So one is, you know about the Chernobyl, the Fukushima, that the nuclear plant, yep. Or like you heard about like some of the things about what else, like some mutagens, something like I forgot the word. Some exposing to the harmful chemicals too. So like a lead, heavy toxic too. They can all damage your DNA structures too. And this will go permanently. And then when the skin cancers and the cancer cell can migrate through your blood, it's called a leukemia. And then goes on to your brain, that's called a brain cancer. Goes on to your other cell, liver, or the internal organs. Yes, it can cause internal organ cancer or internal organ failure too. Okay, why? Again, cancer means that there are cells which is not functioning. It's a malfunctioning cells. So your cells are designed to work, but if that's not working, if your brain cells not working anymore, you cannot think. The same as you get the Alzheimer too, or the dementia. Does it make sense too? Leukemia means what? You have a lack of the red blood cell because cancer cells taking place of the red blood cell, which is supplying the oxygen to your body. What happened to your body then? You always get the lack of the oxygen. So mutation is something we believe that what happened to DNA strands here. I know this is something shown, how many fingers? We have 10 fingers and toes. When the babies are exposing to those kind of chemicals, it's not the UV light specifically, it can be, but it's minimal. Okay, it's more about the chemicals. Why the babies? Think about the baby. Baby starting from the single cell. Yeah, your mother's egg, one cell, and your father's one sperm cell too, 1-to-1. From there, they can be like exploded to what? Many cells too. But during that cell divisions, if something goes wrong in there, something goes wrong like here, damaged DNA or damaged gene, it can cause something like over here too. And there are many like scientific stories about like background story about when the baby's born after the Fukushima bomb, yeah, they get lots of mutation rate. Whether they're born with the no legs, born with the 10 fingers or 10 toes like this too, or born with the legs of the brains too. So that's DNA replication. When there's something going wrong with the DNA here. So we have a few examples of DNA over here. The mutation disease, or we call it genetic disease. Those genetic disease is not really fixable. Once you get it, you have to live with that, and most likely they will have the low rate, the percentage of the survival too, due to many reasons. So one is a sickle cell anemia. So what you see there is that's the normal red blood cells, but sickle cell anemia means that red blood cells are kind of halved. So again, red blood cells are designed to transport the oxygen and carbon dioxide from your body. Now, if that look like this, it's not enough to carry the oxygen, heavy oxygens. So the people born with this anemia, with sickle cell, will always suffer for the anemia, lack of the oxygen. That means that it can be cause lots of the other disabilities too. They cannot run properly, okay? They cannot breathe properly, and they can cause like some respiratory tract failure too. Next one is also known as the have you guys heard about cystic fibrosis? Sometimes known as CF. Yeah, that's on the type of this. Cystic fibrosis is something like shown like here. So what you see is a narrowed blood vessels. That's all because of there are some more or less of the DNA structures there too. Okay, Huntington disease, DMD, or Dutchman muscular dystrophy. So they are born with that kind of odd shape of the bones too. That's all because like they have an extra DNA or lesser DNA or chromosome sometimes. Down syndromes, probably the most famous genetic disease, illness. Yep, that's having extra chromosomes in their body.
I thought it was not enough.
No, sorry, not DNA. Oh, that's a Huntington. Sorry, yeah, Down less. So it's number 21 or something. Okay, so even one change, even a single small change in your nucleus can affect your body like this here. Last one is the frame shift mutations. It's known as a tie shot disease. So I know this kind of sad, but the baby born with that mutation, he or she will suffer with the brain damage. Cataracts born with cataracts. Jaundices, who's showing they go yellow in the color. That's due to liver failure, enlarged liver, and there's a kidney damage, and many. So they will have a really low survival rates in that case too. Okay. These things in now Australia now it's kind of rare. I know we don't really get that a lot, okay, but I've seen a few when my wife gave the birth to my babies. Twins. Okay, so but things happen. So in developing country, which is exposing to lots of the mutagens. So let's have a bit of a mutagen. Oh, we don't have the page here. Later on, we're going to talk more about what can cause the mutations. But what you need to understand for now is even a single missing one pair of that can significantly affect your body. That's something that you need to remember because every cells in your body, you're carrying the nucleus. Every nucleus, you're carrying the same information. If they are not identical, that means they can cause something wrong, like a cancer we talked about today. Okay, I almost got the DNA extracted. The spoon of the DNA. Yep. That's the DNA extraction from the strawberry again. So maybe you're not going to do based on my previous students, your school's not really, I'm not too sure model farms too, they're not really doing the strawberry extraction experiment anymore. Well, due to many reasons, people are having the strawberries in the lab. Oh, yeah, like this. Yeah, or it's not really something useful too, but it's good to visualize how the DNA can be extracted too. It's actually common techniques we use in the lab. They do more in the fine way, but more designed way too. Okay, so that's all about the chapter 1. So we learned about the DNA chromosomes and what the base pairing and then what they're functioning for too. So we started looking into the cells. Any questions? So next week we're going to learn about how the cells are divided using those DNA information there. All good? All right. Let's